So right now, the current mode of testing for SARS-CoV-2 is through reference lab and central lab testing, which means you collect a specimen. It can be out of a car or pretty much anywhere, but then it gets batched with a bunch of other specimens, sent in batches to the reference lab. That takes time, and then the reference lab takes time to build up the batches, run the batches, and all that adds up to days of turnaround time. So it's not quick. And for real-time patient management decisions to be made in hospitals, what's really needed is a test that can rapidly determine status of infection on site when patients are being seen, when they're being admitted, coming in through the emergency room or other channels in the hospital. You know, right now, other conditions that cause pneumonia and sepsis, which are conditions that SARS-CoV-2 also causes, can be easily confused with SARS-CoV-2, but they're right now much more commonly caused by the usual suspects. You know, sepsis could be caused by bacterial infections, severe other viral infections like influenza, pneumonia caused by a whole host of different organisms and infectious causes. What we need now is a way to identify somebody who has SARS-CoV-2 quickly, who presents in a similar way, in a relatively severe presentation, who's going to likely be admitted. So what we've developed is a reference lab quality test that can be run at the point of care in about 45 minutes or less. You take a sample, put it into this new cartridge that we've developed, and about 45 minutes later you'll have the result. And that test can be run at the hospital where patients are being admitted. So the results are available much more quickly. And it means that those results will play into how those patients are managed, who gets respiratory isolation, who needs antibiotics, who doesn't need antibiotics. Those kinds of decisions can be made in real time. I think it is transformative because this is the first time in the world that we've had this capability. They didn't have this capability in China. They had to assume a lot of things about what a patient had when they came in, and so-called presumptive diagnosis of SARS-CoV-2. And that probably put a lot of patients who didn't have it in with patients who did have it, and that's not a good situation. So they didn't have this tool. No country has had this tool until today, right? And so that's what's really needed right now for patient triage and hospital triage of patients who present with severe conditions. People in general are frustrated with the turnaround time that they're getting. They don't know their status until, days later. Knowing your status quickly. If you're a healthcare worker, knowing your status. If you've been exposed as a healthcare worker, knowing your status. This will allow much better decision making relative to deployment of our personnel in the healthcare system. So one of the major advantages that we have is that our systems are already in many, if not most, hospitals in the U.S. They're already there, ready to be used for this test. That's also the case in many other parts of the world. We have over 23,000 systems out there, and many of them are being used for things like tuberculosis diagnosis and HIV diagnosis right now. That installed base can be deployed for use of this test. Same instrument, different cartridge, but same instruments can be used to respond to this pandemic. We think this will be a very important tool in being able to get rapid, actionable results, to let patients know if they're carriers, even if they're asymptomatic, to quarantine, and to know that they're being quarantined for a reason, not because they may have hay fever or some other cause, but really give them a reason for quarantine. And that is very important, to be able to act on the basis of definitive information about your infection status. What's unique about it is the ability to actually carry out all the steps of a laboratory analysis, a reference lab analysis, which means extracting RNA, viral RNA from the sample, amplifying it with molecular amplification methods, and detecting it, All those steps are integrated from the point that you add a sample to the cartridge. And doing that in a seamless, integrated way that delivers a result quickly is what is unique about this. It's very important. I mean, I have two kids who are in healthcare, Alex and Margaret, my adult children in Portland. Alex works in the operating room. Margaret is in frontline patient care. I'm very concerned about the levels of exposure they're going to get. And knowing someone's status, infection status, coming in allows us to be a lot more vigilant about possible exposure. So we'll put the right preventative methods in place, barrier precautions in place, so that we can help reduce exposure. I'm very hopeful that we'll be able to flatten the curve, as we say, by instituting more extensive methods for controlling the infection, reducing interactions by social distancing. These are all likely going to be effective. So if we can flatten the curve, we can minimize the overall effects of this on our hospitals especially. There'll be less strain on the hospital system. It also gives us the chance to allow some of the new therapies to come into play, which are in development, in rapid development right now. And hopefully we can delay this enough to allow those new therapeutic approaches to have an impact.